Hello everyone and welcome to Prof Talks. In today's presentation, we'll discuss the US FDA guidance for industry on submission of quality metrics data. Please note that this guidance has been published a draft version in November 2016 and this has still not been finalized. However, the concept provided in this guideline are very important and can be used as a part of internal key performance indicator assessment by the organizations. And the organizations should be ready to provide this data upfrontly and voluntarily to US FDA. If we look into the content of this guidance, it includes introduction, the background. In background, it provides details for modernization of regulatory oversight and promotion of post-approval improvements and quality metrics data regulatory foundation. It includes reporting quality data and calculating quality metrics, which has who reports and who contributes for the report, quality metrics FDA intends to calculate, quality metrics data needed to be reported, how to submit comments within quality metrics report and how to pose questions to FDA, how to report quality metrics data to FDA. The next part is use of quality metrics and public reporting, how FDA intends to use the quality matrix data, quality matrix reporters list. Then it has Appendix A, which includes the quality matrix data elements, and Appendix B, which includes the examples. Before moving ahead, please ensure that you have subscribed to the channel Prof Talks and have pressed the bell icon so that you can receive the notification of the upcoming presentations. Starting with the introduction. So, quality metrics is used by all the pharmaceutical organization for internal monitoring of the quality system as well as process and drive the improvement. USFDA intends to use this quality metrics data for the same purpose. So, the metrics data can be useful for FDA to develop compliance and inspection policies like the risk-based inspection planning, ability to detect and mitigate the product shortages upfrontly, encourage industry to implement state-of-the-art quality system. Hence, FDA initiated the voluntary reporting phase through this matrix data for limited set of data. Manufacturers submitting this data are referred to as covered establishments, and the products for which they are submitting the data are referred to as covered products. So this guidance is not focused onto the compounder of the drug substances, blood products, vaccines, vitro diagnostic, cells and gene therapy, cells, tissues, and allergenic extracts. Moving to the background part. So the first point is modernization of regulatory oversight and promotion of post-approval improvements. The Center for Drug Evaluation and Research have always promoted a vision of a maximally efficient, agile, flexible manufacturing sector that reliably produces high quality drug products without extensive regulatory oversight. FDA encourages manufacturer to routinely use additional quality metrics beyond that are minimally defined into this guideline for performing the evaluations at site. The quality matrix data, a regulatory foundation. Firms use quality metrics as a part of the product's process validation lifecycle and to the pharmaceutical quality system. So the FDA expects that whatever metrics have been defined into this guidance are included as a part of firm's internal quality management system. Once a control strategy has been successfully implemented, manufacturers are expected to maintain the process in a state of control over the entire life cycle. Manufacturers need to have an ongoing program to maintain and evaluate the product and process data that is related to the product quality. FDA can inspect quality matrix data, but at the same time, they have made it clarified that they do not intend to take action based on the errors in the data submission if the data has been submitted into a good faith. Moving on with the reporting of quality data and calculation of quality metrics. So the first part is who reports and who may contribute to a report. As we have seen into the establishment initially, the covered establishments are the establishments who are manufacturing the product, and the product for which the data is submitted is called as the covered product, right? So the reporting of the quality matrix data can be by the owners of each establishment engaged in the manufacturing or processing of the drug product, API, etc., which include the approved product, the marketed OTC monograph, or the marketed unapproved drug product. Covered establishment also includes the contract laboratories, the contract sterilizers, and the contract packagers. And who reports for the covered establishments? So there can be two type of reports. One is the product report, which is submitted by the centralized product reporting establishment. So FDA prefers that all the covered establishments work together with one centralized product reporting establishment and report the matrix data for the covered product to the centralized product reporting establishment, which will prepare a single report that includes data from all covered establishments. If data is not available for one particular covered establishment, then the centralized working reporting establishment will mention clearly the same. And FDA expects that the quality unit of a covered establishment will be in a better position to provide this data for the quality metrics. Second, each establishment can provide an independent report directly, which is covering all the listed covered products into one single report. The second point under reporting of quality data includes quality metrics that the FDA intend to calculate. Okay, so the first point in it is the manufacturing performance. The FDA intends to calculate the manufacturing performance through a lot acceptance rate. What is the lot acceptance rate? It is number of accepted lot in a time frame divided by the number of lots started in that time frame. For example, if there are 40 lots that are started into 1 year and matrix data is calculated for 1 year, how many of those lots have been accepted lots, which gives an overall manufacturing performance. Second, the FDA intends to calculate the laboratory performance, which is calculated through a matrix for invalidated out of specification rate. So the number of invalidated OOS test results for release and long-term stability testing divided by the total lots released and long-term stability test results in the reporting time frame. And the third is the customer feedback, which is product quality complaint rate. So number of quality complaint received for a product divided by the total dosage units distributed into the time frame. Hence, for the limited matrix data, USFDA intends to calculate the manufacturing performance, the laboratory performance, and the customer feedback. For reporting of the quality data, the third point is the quality matrix data that may be reported. So the preferred data is data related to the lot of drugs that are imported to be imported into the United States or manufactured into the United States. However, FDA identifies a challenge for some of the firms that it may not be always possible for some establishment to identify the started lots, rejected lots, and OS results that are specific to the US. Because some lots are manufactured outside of the US, may be split after manufacturing, and a portion is imported into the United States. So as a way forward, if a manufacturing process uses same process and controls, a report could include data from all lots, that is imported to the US and not imported to the US, specifically for lot acceptance rate and invalidated OS metrics, that is for the manufacturing performance and laboratory performance. The selection of drugs that are either imported and all the drug products that are used into the manufacturing should remain consistent throughout the reporting cycles. So if a firm selects to report data only for the imported products in the US, it should be consistent, or if they are reporting the data for all the drugs product, it should be consistent throughout the reporting phase. Product quality complaint data should be specific for lots imported to be imported or manufactured into the United States. Continuing with the quality matrix data that may be reported, the first point, that is the manufacturing performance, is the lot acceptance rate. So the important points to consider for the lot acceptance rate include number of saleable lots started, number of saleable lots released, number of saleable lots rejected. And it also includes number of lots started for in process and packing product lots, number of in-process and packing product lots released, number of in-process and packing product lots rejected. Specific criteria for the laboratory acceptance rate data includes, if we see the saleable lots, so example of saleable lots are the bulk tablets, filled vials, bulk milled in-process materials, bulk API, and bulk intermediate API. A lot may be subdivided further, and hence each subsequent subdivision or grouping is considered to be as a separate lot. If we see the packing product lots, so example of packing product lots include multiple packing configurations of bulk tablets like small bottles, large bottles, blisters, and labeling filled sterile vials with multiple labels intended for different countries. So FDA anticipates pending disposition lots, example, the work in progress, lots evaluated for batch release, lots pending disposition due to quality related discrepancies is equal to number of lots started minus the lots released plus lot rejected. There are rare instances when this construct will not be valid. For example, lots pending disposition for an extended period. Use of a comment text box to explain occurrence of such an anomaly. The second point for the quality matrix data, that is invalidated OOS rate. So for calculating this matrix, the information needed is the number of lot release test OOS and long-term stability OOS results for finished drug product or API, number of total lot release test and long-term stability test conducted, number of OOS test results for lot release test and long-term stability test that were invalidated, and the specific criteria. It states that investigation must be conducted whenever OOS results is obtained. For quality metrics, the following OOS should be counted: finished drug product and API and long-term stability test results only. All finished drug product and API and long-term stability test that initially indicate OOS, even if source of OOS is investigated and determined to be an aberration of the measurement process, that is, it is invalidated. Number of test results is a measurement tool that provides context for invalidated OOS rate and provides a secondary matrix for manufacturing performance and ability to product within limits. Lot release and long-term stability OOS results investigated as a manufacturing aberration divided by the total number of lot release and long-term stability tests performed in the same current reporting period. Continuing with the specific criteria for invalidated OOS rate, the tests include a single analytical result for lot release or stability time point with an established limit. Example, the analytical chemistry, release sterility test, etc. The example includes for lot release, final content uniformity results as reported on the COA is considered as one test. For a stability time point, each test performed in the time point would count as an individual test, and covered establishment that cover manufacturers APIs used in covered drug product is not expected to report the stability OOS. So this is a very important point. For stability testing, only tests that support real-time stability of product should be counted. That is, accelerated stability testing is excluded from this reporting matrix. If lot release for long-term stability test is conducted multiple times for a lot, for example in the case of retest, each test should be counted. Other type of testings like the in-process testing, environmental testing, raw material and packing component testing should not be counted in this report. The third quality matrix is the product quality complaint rate. So the requirements are the number of product quality complaint received for the product, the total number of dosage unit distributed for the product. The specific criteria include total number of all product quality complaint does not include multiple counting of the same product quality complaint if the receiver forwards the complaint to the individual manufacturers for investigation. This number does not include all potential quality issues like the lack of effect, which is more of a pharmacovigilance type of a complaint. The total number of dosage units distributed for the product family to customers including the distributor. So reporting of quality data and calculation of quality matrix. The next part is how to submit comments within quality matrix data report and how to pose questions to the FDA. So the establishment may submit a 300-word text comment to provide an explanation of the submitted data or report plans for improvement. Example of a comment can include an unexpected decrease in the lot acceptance rate may be due to a situation outside the control of facility like the act of nature such as storm or fire. Comment can also be on the new technology or other improvement methodology adopted. Any other specific questions can be sent to the FDA on a specific email ID mentioned here that has been created by FDA in the name of Quality Matrix. Then, how to report the quality matrix data to the FDA? So the reporting establishment may submit quality matrix data report where the data is segmented on a quarterly basis throughout the single calendar year. And FDA expects to begin the analysis when the portal is closed for a specific year and then publish the initial findings and quality metrics reporters list on the FDA website. Moving to the next part, the use of quality data and public reporting. So, how FDA intends to use this quality data? FDA intends to use the data for focusing resources onto the high risk areas. They intend to establish signal detection program in identifying establishments and product which possesses a significant risk. They intend to identify situation where there is drug supply disruptions, improve the effectiveness of the inspection program, improve FDA's evaluation of drug manufacturing and control operations. FDA foresees a challenge, that is, this exercise will be limited if a large amount of data is not received into this voluntary receiving phase. Hence, the data may not be representative. Also, self-selection bias may increase the risk of signaling an outlier when there is none. So FDA will use the information collected to focus on working with establishment for early resolution of the quality issues and hence clearing off any disruptions into the product supply, helping to prepare and direct for the inspections, using calculated metrics as an element of post-approval manufacturing change. FDA intends to publish the data analysis on the website. What agency has learned, how it has impacted, frequency of GMP inspection, and how it has addressed potential drug shortages. The second part is the quality metrics reporters list. So FDA intends to publish name of all the organizations sharing the metrics data voluntarily, as demonstrates a commitment to increasing transparency between industry and the FDA. So this may be useful for establishments when selecting the contract manufacturers or component suppliers based on the list of data, which can give an assurance about the performance of that particular organization. And it can be also helpful for the healthcare purchasing organizations for selecting the consumers in sourcing the drugs. Categories for identifying the organizations. So product reporting establishments, we have seen that one centralized agency will submit the product report. So product reporter top tier. So this will be one reporting category. If complete data supporting all metrics was submitted for a particular product by the central agency for a year, that is, if there are 10 manufacturing establishments that have that are identified in the supply chain for that product and data is available for all the 10 organizations, then that product is the top reporter top tier product. Product reporter mid tier. If all covered establishment in the supply chain were identified in the report and data for at least one establishment for each product was shared for the full year, then that is the mid tier. And product supply chain reporter, like all covered establishment in the supply chain were identified in the report. The second part we know is the site reporting establishments. So site reporter top tier. Complete data supporting all metrics included for all covered products for the full year has been provided by the site. So that is a top tier site. And mid tier, if complete data supporting all metrics included for at least one covered drug product manufactured at an establishment for the full year has been provided. If we look into the examples for the reporting categories for product, so if product reporting centralized organization, for example, if there is some ABC product reporting establishment which reports identifying all covered establishment in manufacturing supply chain, then but it did not provide quality matrix data, so ABC would have a product supply chain reporter designation. It provided matrix data for primary manufacturing establishment for each product, but incomplete data was submitted from other establishment. Then that product reporting establishment centralized agency would be a mid-tier designation. And it has submitted a complete report of data listed above for all covered drug products. Then it would have a top-tier designation. Same for the case of site. If there's site XYZ which manufactures different product and they have at least covered drug products and establishment and data supporting all metrics, then for one product at least, then it would be a mid-tier designation. And all metrics for each drug product, then it is a top tier designation. So one important point for site reporters is that if the site reporters have opted to send the data to a centralized product reporting establishment, then if they have provided all the metrics data, then also they will have a top reporter establishment categorization. Now we move to the Appendix A, information, identifying the information and quality matrix data elements. So the information is separated in 8 subparts corresponding to the different combination of report, establishment and product type. So if we look into the product report and the applicant part, so the first part is product report, application product, finished drug product. Second is product report, application product, API. There is then non-application product. So under non-application product, again there is finished drug product and API. For the site report, there is manufacturing with product quality oversight responsibilities only, manufacturer with testing responsibilities, manufacturer without testing responsibilities, and manufacturer with testing responsibilities only. As seen in the previous slide, the first subpart is application finished product. So this is just an example of how the data can be. And if you can see into the details, here there's a product reporting establishment, that is this, with oversight responsibilities only, contract manufacturer performing release or stability testing, contract manufacturer not performing release or stability testing, and contract laboratory performing release or stability testing only. And if you can note from here to the part here, this is all the generic information, that is general information about the drug product, and the highlighted part here is the matrix data. So the matrix data include starting of the in process or packing lots, starting of the saleable lots, rejected in process packing lots, rejected saleable lots, rejected in process packing, rejected saleable, number of quality complaints, number of units distributed. Then there is details related to the OOS information and sum of all release and stability test. So this is the information that you need to give for the specific particular product. The second subpart, that is application API, same information that we have seen for the application finished product is also applicable for the application API part. So here, if you can note, the details for the matrix are available here, and up to this point there's general information. So this information is same and details are to be provided into this format. The next part is the non-applicant finished product. Again, this is the same information that we have seen into the previous two parts, that is application finished product or the application API. So this is non-application finished product and data remains the same. And then again, non-application API. So this again, information provided here is the same for the part of the matrix. Out of the 8 subparts, we have seen the first 4 subparts related to the product report. Now, moving with the site report, the site report consists of additional 4 subparts. The first part includes manufacturer with oversight responsibilities only. So if we can look into the matrix data, and if we can look here, so it includes the finished product application, finished product non-application, API application and non-API application, and the information till here is the general information, and the details from here till the end are the matrix related data. So these details are to be provided for site report for manufacturers with oversight responsibilities only. The second point for the site report is the manufacturer that perform the testing. So the left side criteria, that is finished drug application product, finished drug non-application, API non-application, remains the same, and the data that has to be provided for the matrix also, it also remains the same. It is only the details for the manufacturer that performs the testing. The next part is the manufacturer that does not perform the testing, so the data matrix and the details remains the same. And the last, that is manufacturer with testing only. So manufacturer which performs the testing only, so information is same. Moving to the Appendix B for examples, the first matrix part is the lot acceptance rate. So here are some details for the lot acceptance rate. For example, if 6 small in-process lots are combined into 1 saleable lots, and 2 saleable lots are then combined into 1 packing lot, and if all the lots that are started are released, then the first criteria, that is in process and packing lots started and released, will be 6 lots uh, for each in process lot. And now we know that 2 in process lots will be combined into 1 saleable lot. So 6 plus 6, then it will be 12 plus 1 packing lot, so the total amount comes out to be 13. And saleable lots started and released, so 2 saleable lots will be combined into 1 packing lot, so saleable lots will be 2. One saleable lot manufactured, that is separated into 5 packing lots. So if there is one saleable lot and it is then segregated into 5 packing lots, so in process and packing lot started and released will be 5, and saleable lot started and released will be 1. For example, for the OTC product, one batch of a saleable product is packed into an unlabeled primary pack. Okay, and primary pack is labeled and placed into secondary packing in 3 different packers. So there are totally 4 sites involved. One is where the batch is manufactured and packed into unlabeled primary packing, and then that primary pack is labeled and placed into secondary packing at 3 different packers. Then all 4 facilities are the covered establishment, right? For unlabeled primary pack manufacturer, the unlabeled lots are the saleable lots, while the lots which are distributed by each packing establishments are the saleable lots. Facility A manufactures product and Facility B packages the product. Facility B discovers a defect that leads to rejection of the lot. Defect was due to the manufacturing at Facility A. Then Facility A should not count this product lot as a released lot in spite it has been released earlier. For Facility B, if defect was discovered upon incoming acceptance testing and packing lot was not yet started, the lot should not be counted. If a packing lot was started, it should be counted as lot started, not as the lot released. Continuing with the lot acceptance rate, for non-functional or functional coated tablets where coating process consists of multiple separate coating pan loads, then the count of lots depend on whether separate pan loads are considered unique lots or if loads are part of a single started lot. For either functional or non-functional coatings, samples collected and tested for finished drug product release should be representative of the lot. Now, for example, if a facility A initiates manufacturing of product Z in the last quarter of the reporting cycle or stops the manufacturing of product Y in the first quarter of the reporting cycle, an explanation of partial year can be described in the field comment. The product report or site report would be considered complete for that product. The Appendix B example for the product quality complaint rate. So if a lot is distributed to 5 customers and all customers report the same complaint, then this should be counted as 5 complaints. If a lot is distributed and single customer submits the same complaint from different department, only a single complaint would be counted. A lot is distributed to 3 regions and complaint is received on the lot from a region which is not into the US, then this metrics data need not be reported because the metrics data is limited to the products that are intended for the US market. For a site report by a packager, if a complaint is received and potentially due to packager's operation like the discolored tablet or powder, the complaint would be counted by the site reporting establishment. Appendix B, third part, the invalidated OOS rate. Now, if an analytical test with multiple sample preparation or injections to generate a final result, one test is represented by a single analytical result with an established limit. For example, one content uniformity test proceeding to stage 2 may have 30 invalidated results. Only one OOS result would be counted. If 2 samples from one lot are tested with 2 injections each and there is one result reported on the COA, this is considered as one release test. If an OS result occurs during in-process testing for a test that is considered a real-time release test, this is considered a release OS result for the purpose of this guidance. If more than one OS result is observed during finished drug product testing, example, the lot fails both assay and uniformity, this is considered multiple release OS results. If, for example, if a 50 kg of API is packed into 5 10 kg packages and 3 to 5 of the containers are tested, COA reports the average. So if one or more container result is OS for same attribute, establishment should initiate an OS investigation and count this as a single OS. Single API container with OS results should result in investigation of the lot in its entirety. After investigation is complete, subsequent retesting should be counted as new release test. Now, if a company A does not declare OS results until initial laboratory investigation proves the result is valid, and if the results are invalid, an original result is not labeled as an OOS, there will be no record of invalidating this OOS result. Thus, this will lead to a lower invalidated OOS rate for the company. Hence, the guidance requires lot release OOS results should be counted prior to the laboratory investigation. The types of results should be evaluated as a part of the product review to determine the need of change in the drug product specification or manufacturing or the control procedures. With this, we come to the end of this presentation. Hope you have liked this presentation. Kindly subscribe to the channel Prof Talks to get more information onto the pharmaceutical quality system. Thank you.